Well, thank you everyone for joining. We will wait for a couple of minutes to allow for all participants to join, and then we will start right away. Okay, well, it's almost 12:01. We'll wait for a minute for allow everybody to join, and then we will start promptly. Okay, let us start. So thank you everyone for joining us, and welcome to the 2022 James E. Dalen Distinguished Lecture for Health Policy. Launched in 2008, the James E. Dalen MD and PhD Distinguished Lecture for Health Policy was established to commemorate the legacy of Dr. Dalen and his countless lifelong contributions to the advancement of the University of Arizona and his key role in establishing the Mellon Enid Zuckerman College of Public Health. This annual lecture is a forum for high-level national and international leaders to share their knowledge and perspectives on issues related to population health and policy. The goal of the series is to deepen understanding and raise awareness of public health issues, as well as to connect the academic world with the real world of public health. So thank you, Dr. Dalen, for establishing this series. Today, we will be hearing from the chair of the Department of Pediatrics at the College of Medicine in Tucson and the physician-in-chief at Banner Diamond Children Medical Center, distinguished colleague and dear friend, Dr. Fayez Ghishan. So it is my pleasure and honor to introduce Dr. Fayez Ghishan. Dr. Ghishan is a board-certified pediatric gastroenterologist at the University of Arizona College of Medicine, Tucson, and ranks as an internationally recognized expert in gastroenterology and related clinical research. His laboratory has been in the forefront of research on the microbiome and nutrition in children, work that has resulted in hundreds of publications and numerous awards. His research has been continuously funded by the National Institute of Health throughout his more than 40 years career. He has received 2 prestigious merit awards from the NIH, one in 1996 and a second in 2008. Among the many honors and awards he has received over the years, Dr. Ghishan was the first pediatric gastroenterologist to be awarded the Horace W. Davenport Distinguished Lectureship in 2014. This is the most prestigious recognition by the gastrointestinal and liver physiology section of the American Physiological Society. Dr. Ghishan was recruited to the University of Arizona as professor and head of the Department of Pediatrics and director of the Steele Children Research Center in 1995. He was previously director of the Division of Pediatric Gastroenterology and vice chair for the research at Vanderbilt University. He is a distinguished alumni of the University of Iowa and Pennsylvania State University and has been an invited lecturer at numerous universities, both nationally and internationally. It is my honor to welcome you, Dr. Ghishan, for this series. Thank you very much for being here with us.
Iman, I'm so delighted and be honored that I'm joining you in delivering the Jim Dalen Lecture in Public Policy. It is my great honor to basically be your friend And we've been together for so many years at the University of Arizona. And I'm so delighted that the audience are from various disciplines, including the College of Public Health and the College of Medicine. So I'm going to start by showing you my talk. First is the microbiome and health and disease. Just to let you know, my lab is the core facility for the entire University of Arizona and doing second-generation sequencing. So we sequence everything you could think of, microbe, viruses, for example, yeast. And recently I added a new instrument to our center, and that's called the MySeq 6000. And that machine can do whole genome sequencing, which we'll be offering in the next month or two. And that does entire human genome in 19 hours. Remember, I want to tell you, in 2003, when President Clinton announced sequencing the human genome, it took 6 months to do one human genome at a cost of $100 million. Today, in my lab, we'll be able to do sequencing of the entire human genome in 19 hours. Our cost will be about a couple of $100. So imagine how the technology has evolved. But let me first share with you my story with Dr. Jim Dalen. So in 1995, I was invited to come and look at the chair, and the dean was Jim. And he is clearly the most intellectual person I have ever met in my entire life. Within minutes of me sitting in his office, he literally offered me the job to be the chair of pediatrics here. He's not only was the dean of the College of Medicine at that time, and he stayed for 11 years as a dean. He was a mentor, he was a friend, he was clearly a clinician and scientist. And the best thing about him, he is really the best storyteller ever. So I want to tell you a story about Jim. So we introduced to a ladies in Phoenix, they call themselves Panda. People Act Now, Discover Answers. So Jim and I, we went to talk with these ladies in Phoenix at a really nice hotel. And I can tell you the asset of a dean. So Jim basically started telling them a story. And it was absolutely fascinating story. And everyone was really excited. And then he said, ladies and gentlemen, this is the reason why I'm here. I'm here as a dean because I want to introduce you to this new physician scientist who's going to lead our pediatric research. These ladies so far has raised more than $15 million because of the Jim Dalen taking me to meet these ladies in Phoenix, convincing them about doing pediatric research. In fact, all the equipment I have in my lab belong to these ladies. The sequencer, it's 1.3 million, the NovaSeq 6000, $1.3 million was given to us, again, because of what Jim has done. And I can't thank you enough, Jim, for your leadership, for being a mentor and a friend over the years. You and Priscilla has been our friends for so many years, and I'm grateful that we got to know you, and I came into Tucson, Arizona. Because of you, I came into this institution. So let me tell you what I do in the space of microbiome now. So first I want to show you, this is the tree of life. Why I'm showing you this, because it's clear, as you could tell, we are here as vertebrate animals, but the bacteria and archaea, as well as fungi and plants, we belong to the tree of life. So we are connected. And therefore, when we talk about the microbiota, I want to emphasize to you, microbiota is the collection of microbes in our GI tract. Microbiome is the collection of genes encoded by the bacteria. Okay. So as you could tell here that we have a skin microbiome, we have oral microbiome, we have digestive disease microbiome, and we have urogenital. But the truth is, we have also viruses live within us. Archaea is less than 1% of the genome. Okay. But we still have archaea, we have fungi, and we have parasite. which live within us. So we are superorganism. And in fact, what we do in our lab, we do 16S ribosomal RNA, and I'll show you the equipment we do. That will give us basically the bacterial flora. Metagenomic, we can do it at the new sequencing, we call it NextSeq 550. That will give us genome analysis of the entire microbial community. Remember, 20% of the bacteria in our GI tract can be cultured. So what happened with the 80%? That's what the technology is. The technology is doing sequencing using these Illumina sequencer for analysis of our gut microbiome. Now, natobiotic mice, I'll mention something about natobiotic mice in my research. And those are animals which have devoid totally of bacteria. We use them as a model. We call it germ-free animals. And we introduce to the animals, let me tell you, start with one experiment we just did. We took stools from a Crohn's patients and we introduced it into the GI tract of these mice. These mice immediately, within days, they develop colitis, suggesting that clearly the microbiome in patients with Crohn's disease plays a major role in the development of symptoms of Crohn's disease. However, just to understand the biological classification, I put this to show you that we start with the kingdom of bacteria and we go down to the phylum, subphylum, class, all the way down to the species and the strain level. We are able now to give you strain level by doing analysis in our lab. So one single bacteria, we can know exactly what bacteria, for example, is causing what in a patient with, let's say, autoimmune diseases. So it's clear now we live in harmony with the bacteria. And this is the cover of Science magazine suggesting that the gut is the really, truly the inner tube of life. And I'll explain to you why is that, why we do that. So there is connection between the brain and between the GI tract. When I tell you that I have a gut feeling that this basically story is correct, well, that's absolutely true because you have literally a brain in your gut and the brain in your gut is connecting with the brain in your head. In fact, when you think about it, there is more enteric nervous system in the GI tract more than your entire spinal cord. So it's clearly the connection between metabolite of the bacteria and between hormones and cytokines between the brain and the gut. So if I tell you that a patient has, for example, SIBO, small intestinal bacterial overgrowth, many of these patients, they get what we call foggy brain. Why? Because the bacteria in the gut produces metabolite, and that metabolite basically causes the problem. That is in the setting of small intestinal bacterial overgrowth. So this is a basically structure of, I'm showing you this because I'm going to show you some slides actually with the gut. Look, the gut and the skin are the two most highly productive organs in your body. Every 5 to 7 days, you renew your GI tract. All this renewal comes in from stem cells in the crypt cells. And every 5 days, you renew yourself. It's estimated that during your lifetime, you will shed 1 ton of epithelial cells throughout your lifetime. This is a cut section at the tip of one of these villi. I want to impress on you that this is what we call the microvillus membrane. This is one single cell. This is capillary with red blood cells beneath the basolateral membrane, ready to transport nutrients to your liver. So, however, it is clear there is a space between this lining of the microbe and between the mucus layer and the bacteria. You don't see it here, but I'll show you a slide to show you exactly what I mean, that this is what we call a eubiotic state, that is a healthy state. So we, as a human, we are superorganism. We have 10 to the 14th power of bacteria in your gut, 100 trillion, literally. We have 10 to the 12 cells in our body, and we have 10 to the 11th power of stars in our universe. So clearly we have more bacteria in our gut than the cells in our body and than the stars in our universe. But we have to live in harmony with this bacteria. So over 100 trillion organism has been described. 100-fold, we have more genes in our gut than us. And our flora is part of integral of our genetic landscape. So this is just to illustrate to you, the gut microbe had 3.3 million genes. Our human body has only 23 genes, 23,000 genes, that's it. So actually, when you think about it, the human DNA we are only 10% of our DNA is a human, and 90% of our DNA is bacterial. It's not a humbling experience to think of us as only 10% a human because we have only 10% basically DNA from a human. Okay. Nonetheless, the important point, we have to live in harmony with this bacteria in our gut. Where do we get this bacteria? Well, now we believe that there's some bacteria present in the amniotic fluid. It used to be thought that amniotic fluid is sterile. That's not true anymore. Phase 2, you acquire it from the vagina, from the mom gut microbes through the feces, and from the hostel if the baby is born by C-section. Next is breastfeeding versus bottle feeding. Breast milk is the best thing ever created by God. The bifidobacteria dominate 90% of our flora, and that's really the probiotic. Best probiotics ever created is the probiotics driven by our breastfeeding. Why is that? Because the mom has something called HMO, human milk oligosaccharide. When it is bottle fed, there is more bacterials and Clostridial species. And by 2 to 3 years of age, we acquire the adult. So I want to introduce you to the new topic now. We believe there is a critical window in babies' microbiome, and that's the first 1,000 days, because after 1,000 days, you acquire the other bacteria. During that window, if we are able to have a good microbiome, or i.e. diverse microbiome with a bacteria, your risk of having inflammatory bowel disease in the future is significantly lower. I can prove it to you by animal models, which we have done, okay? But nonetheless, the important point here, critical window of having a diverse microbiome in the first 2, maximum 3 years of life. So vaginal delivery is much better than C-section. Why? Because when the mom is pregnant, it changes her gut microbiome. The mom changed her skin microbiome to allow the baby to latch on the breast. It changed basically the skin microbiome as well as her vaginal microbiome, and her gut microbiome also changed. And that's the reason why the vaginal delivery is so much better for babies compared to skin microbiome actually acquired when you are C-section from the mom's skin and acquired from the hostile microbiome. So nowadays, there is a really new concept which to take basically swap of the vagina from the mom in a baby or breast who are section is born by C-section, and basically swap the baby to allow the baby to have this diverse microbiome as seen here by the color red as well as blue. Okay, so this is exactly what I told you. Birth dictated by pregnancy, healthy pregnancy, mode of delivery, and then by 2 to 3 years of age. The bacteria, for example, change to adult bacteria after the age of 3 years. Now, we do animal work. So people ask me and say, okay, Dr. Shah, you do animal work, for example, in the microbiome. However, how that's relevant to humans? I can assure you, this is the red color is the mouse microbiome, and the blue color is the human microbiome. The difference is only 15%. So 85% of the microbiome of the animals, the mice in this situation, similar to what we have in humans. Okay, so I want to introduce you to the new concept now. Whatever you do in life, you have to do it persistent. If you change your gut microbiome, if you start eating a Mediterranean diet, in 2 weeks' time, you'll change your gut microbiome. However, if you go back again and start eating Western diet, fast food, microbiome will go back again. If you don't hear anything from this lecture, I want to tell you one concept here, that lifestyle and diet dominate over genetic in shaping your gut, shaping your health. So biology is not your destiny. Your destiny is dictated in your hand. Whatever you do, lifestyle, exercise. Exercise is medicine. Whatever you do, exercise and eat healthier diet, That will change your gut microbiome. So it's clear to me, whatever you do in life, you have to be persistent. Okay, so when did this revolution happen? 2006. Okay, so this is the equipment in my lab. 20% of bacteria can be cultured, but 80% we do this. This is a MySeq, which does 16S ribosomal RNA. It gives you basically a species level. However, the next seq will give you strain level. We are able to tell you the strain level. This is done by this equipment, which is available in my lab. And incidentally, we do it in collaboration with anyone who wants to collaborate with us for no fee. However, if you want to do it for yourself and independently, we charge because it's a core facility. And the MySeq will be next to this. And unfortunately, I can't show you the picture here because it will be delivered in the 15th of April. Okay, so my feeling is this. In a healthy state, you are in a healthy state. You are the host. You have a tolerance and harmony with the microbe in your gut. That's called eubiosis. You need to be in this equation here in a healthy state. Dysbiosis is a bad state to be in. This is you have a breakdown in tolerance between the microbe and host. Why do I know this? If you take patients with Crohn's disease, they make antibodies against normal bacterial flora in their gut. Okay. All of us, we eat bread. How do you make bread? You eat bread because you make it with yeast, and we have yeast in our gut, but we don't make any antibodies against yeast. Okay. So if you have a breakdown in yeast, you have anti-Saccharomyces cerevisiae antibody against the Saccharomyces, against the yeast in your body. That's not a healthy state to be in. Okay. So this is a work we have published with our collaborator in Sweden. Okay. This is published in a journal called Gut, this British journal, one of the most highly cited paper I have written. Okay. So this actually is biopsies of patients who have ulcerative colitis and control. Okay. So this is the line, this is the healthy control. Blue color is the lining of your gut. The green is the mucus. The red is the bacteria. You see the space? And I showed you electron microscopy before to show that there is separation. We measure the space literally between your gut and between the mucus layer. Okay. Now, this is a patient with ulcerative colitis, but treated with Remicade, and he is in good control. He's in remission. Beautiful. Oh, this is Mayo 1, i.e. moderate ulcerative colitis. Look at this. Actually, the space is so narrow, basically. And some of the bacteria actually overlying the gut. This is a severe ulcerative colitis. You see, there's almost destruction of the epithelial lining. Okay. So why I'm showing you this? Because I'm going to tell you in the end of this talk what sort of diet you can eat to have this basically space, because there's certain food which I'll tell you about, and that will give you this type of picture. Okay. So that's a dysbiotic state. That's another healthy state to be in. Okay. Okay. So is your gut microbiome similar to mine? No. Each one of us has this different microbiome. Okay. So actually, the microbiome is like a fingerprint. Okay. So each one of us has a different microbiome. Okay. Okay. So now I'm going to tell you some of the work we do. And I'm taking example here of when we have inflammatory state, what happened to you. This is a review article we have written with Pavel Kela, my collaborator in my lab. So we have discovered that actually when you have a diarrheal disorder, you inhibit some of the transport proteins, such as sodium hydrogen exchanger, for example, in this case. Okay. So we basically have a mouse model in which NH3, the major transport protein in the GI tract, which is knocked out. Okay. Oh, look what happened. Okay. So in this situation, this is NH3 mice. You see the blue color here is interferon gamma, significantly interferon gamma. Why I know? Because we use for a cyalo GM1, which is inhibitor of it goes down, all the way down. Okay. So iron NOS, basically inducible nitric oxide, is markedly elevated in this setting. Okay. So this is a basically healthy control mice, which absolutely normal sodium hydrogen exchanger. Okay. Look what happened to the epithelial integrity. So we have what we call occludin, ecadherin, actin, which hold our cells tight together. Okay. So this is control. Look at this, beautiful here. Okay, the red color is the adherent in between the cells, hold the cell tight. This is active. Look, here when we knocked out NHE3, basically total disruption. See him mildly stained, basically actin. Here, totally misplaced, for example, EK adherent. So there is a leakage of the gut. How do I know this? We do electron microscopy. You see here, knock down NHE3. Look at this, wide elevation, wide marked, basically space. And this is marked space. Here, look at this, tightly paracellular pathway, very tight. So what we discovered in these mice, that they have basically developed spontaneous colitis. This is a picture, for example, of colitis. The red color here, periodic acid stain of goblet cells. See, goblet cells totally gone, okay? Totally gone. So we did the microbiome of these animals, okay? So the microbiome, as you could see here, distal colon, as well as distal colon mucosa, this is the content. See this wild type, and this is the diversity goes down significantly. So the microbiome get changed. So the message here, microbiome change when you have inflammatory state, okay? So this is basically phylum level analysis, NHE3 knockdown. See, totally different than the wild type. In fact, if you look at the distribution of the microbiome here, similar exactly to Crohn's disease, which is inflammatory state, okay? So I'm showing you here for the first time ever that we are showing that these epithelial sodium hydrogen exchanger that will allow us to absorb salt, they are not only simply for absorbing salt, they are involved in the integrity of the GI tract and maintaining diversity of your GI tract microbiome. Okay, we extended this work, and this is still in preparation, to look at cancer cells. This is basically, we went to pathology department with our friend A.B. Bhattacharya. He gave us basically samples. We looked at NHE3 expression in cancer versus normal. See, markedly decreased. This is one patient versus another patient. You see, normal here, and it's way down. So sodium hydrogen goes down significantly with cancer as well. Okay. So what I showed you from this experiment, which we have done, that loss of the sodium hydrogen exchange activity lead to spontaneous microbially driven distal colitis. And then NHE3 activity is required for the maintenance of diverse microbiome. So I'm tying here basically infection, inflammation with shutting down NHE3 with dysbiotic state. Okay. So you need to stay out of the inflammatory zone. Okay. Now, what we have done, one more experiment in this setting, we took basically fecal transplant, FMT, fecal microbial transfer from these animals and introduced them into germ-free animals who are having AL10. AL10 is anti-inflammatory. Knockdown might resulted in accelerated and exacerbated colitis. So I'm confirming to you now, in animal models, as well as in the human models, if we take microbial flora from someone with inflammatory state, you put it in a germ-free animal, you can induce, basically decrease in the diversity of the microbe, and you induce inflammation in these animals. So it's clear our microbe is very important for us in a healthy state, but in a disease state, when you have inflammatory state, actually you will have a dysbiotic state. So one more time, diet and lifestyle dominate over genetic in shaping your microbiome. I put here a picture for you of all the disorders were associated, all these disorders associated with change in the gut microbiome, mostly neurodegenerative disorders, okay? Diabetes, for example, obesity, asthma, for example, IBD, rheumatoid arthritis, all those are basically have change. So we have some preliminary data in our lab to try to explain why women have more autoimmune diseases compared to men. So we have given, in animal models, given antibiotic for men, for a male mouse versus female mouse. And the change is enormous between woman or female and between a male mouse. Same thing, we have published a new work in microorganisms, just published a couple of months ago, to show that adult versus young. If you take an adult antibiotic, it changes your gut microbiome, it takes you a year to really recover it back. That's maybe one of the reasons why adult patients, when they get in trouble with antibiotics, they get C. diff compared to kids. They get less C. difficile. Okay, so this is the role of diet. Okay, so remember, if I give you a fiber-rich diet, you remember I explained to you the space. Space is wide open, okay? If I give you fiber-free diet, see the space goes down significantly. So this is a message that we need to eat healthier diet which have fiber. How do you get fiber from? Mostly fruit and vegetables. We need to eat fruit and vegetables with its skin, okay? That's very important for us, okay? Next, artificial sweetener. I want you to stay away from artificial sweetener. This paper just came out in Nature, okay? Basically using a non-calorie artificial sweetener, okay? It changes your gut microbiome, okay? It makes you actually go into a metabolic state of the obesity. You will gain more weight actually if you consume a lot of artificial sweeteners. Okay. Next is emulsifier in your diet. Stay away from emulsifier in the diet. What's emulsifier? Emulsifier are detergent like soy lecithin, for example. How do you mix, for example, olive oil with vinegar? You have to add detergent to it. And what detergent is they added? Soy lecithin or carboxymethylcellulose. Okay, there is new evidence now to suggest that actually emulsifiers are harmful for basically the mucus layer. It becomes very thin. I'll show you some slide on that. Okay, look, this is basically given emulsifier in the diet. Look at the thickness of the mucus if you give water alone. Look at the mucus thickness in carboxymethylcellulose. Look at the same thing, basically P80. And again, the space is almost gone, literally. And the bacteria become approximate. So you need to stay away from artificial sweetener. You need to stay away from low fiber diet. You need to stay away from basically artificial sweetener in your diet. And next is maltodextrin. Maltodextrin is a filler, has no nutritional value, period. Okay, look at this. This is the experiment in which you have animals were taken no maltodextrin, for example. Look at the space between the mucus layer and between the epithelial lining. Look at this here, maltodextrin, for example. It's way decreased. So stay away again from maltodextrin in your diet. Okay. So there is a new, basically, biomarker for coronary heart disease and kidney disease. That's called TMA, trimethylamine. If you eat a lot of red meat, for example, which has a lot of choline and carnitine, the gut microbiome basically will change the choline carnitine to trimethylamine. Trimethylamine get oxidized in your liver. It's called TMO, trimethylamine oxide, and that causes atherosclerosis, strokes, heart attack, and so forth. So what inhibit basically lyases in your gut? Extra virgin olive oil. So you need to use extra virgin olive oil as well with your diet. Okay, the final thing I want to show you some more data from my lab to connect basically epithelial sodium hydroxide, a new one we cloned, it's called sodium hydroxide 8, with gut microbiome and inflammation and colon cancer. Okay, so we knocked out this sodium hydrogen exchanger. Just got published a couple of years ago. Okay, you see here, decrease in PS stain of goblet cells, inflammatory state, mucus layer here, well organized, here totally misorganized. Look at the space again. See the space I mentioned to you before? This is control, beautiful space. Here, narrowed space when you knock out sodium hydroxide 8. Okay, so we took ulcerative colitis patient as well. The red color is sodium hydroxide 8. See this? Beautiful. Here, totally gone, ulcerative colitis patient. So again, I'm emphasizing to you inflammation, okay? So this is in colon cancer, okay? You could see here normal intestine, tumor tissue. See the red color here, sodium hydroxide, totally gone, totally gone, totally gone. Now, this is the important point. We looked at number of polyps in these animals. See, this is knockout. See, these are polyps, precancerous polyps. This is wild type, healthy control, no problems, okay? When Jim Dalen was my dean, he basically introduced me to this alternative medicine. So I basically joined with Andrew Weil, and we got 5-year award from the NIH. So what I'm going to study as a scientist, you know, I mean, so I studied basically botanicals, which have anti-inflammatory activity. People think has anti-inflammatory activity, but it's not proven. So this is one of my papers. We looked at basically curcumin, okay? Curcumin is the active component of turmeric, okay? Look at this. This is animal models of colon, basically polyps, okay? And I gave them basically 0.5%, it's totally gone. Polyps are gone. So I want you to, in the end of the day, spice your life with little curcumin, because clearly, I don't have time to show you all the data, but it's clear it inhibits NF-kappa B, okay, which is very good for you. Okay, so people ask me about probiotics. Do I use probiotics or not? Okay, so what are probiotics? Pro for biotic life. The definition, the scientific definition, live or microorganism, which when given to adequate amount, confer health benefit to the host. Okay, so I want to assure you that probiotics does not work when you have inflammatory state, period. Why is that? Because probiotics, like you and me, like to live in a nice environment, like to live in a foothills, doesn't like to live in a place where there's so many toxins, so many inflammatory state. If you have a patient with Crohn's disease, ulcerative colitis, or whatever, rheumatoid arthritis, they have inflammatory state. They have dysbiotic state. What you do is you allow the patient to go in remission by treating the patient, and then you give them probiotics. However, probiotics does not work unless you take prebiotics with it, because the bacteria loves to eat, same thing like us. What's a prebiotic? Prebiotic is the fiber in your diet. Okay, so every cell in your body uses glucose. In the GI tract, the intestine uses glutamine, the colon uses short-chain fatty acid. Where do we get short-chain fatty acid? We got short-chain fatty acid from the end product of probiotics or bacteria in your gut, fermenting fibers. One of the best fiber I tell my people is the skin of the banana. Skin of the banana, don't throw it away. That has very rich prebiotics. Okay, prebiotics with probiotics in a healthy state will work very well for you. Okay. So how do I know these probiotics work? Okay, so I published this work in JCI a couple of years ago as an editorial to look at something called Lactobacillus rhamnosus LGG. LGG is Golden and Gorbach, 2 microbiologists in Boston, described this probiotic, we call it now probiotics, in a healthy 32-year-old man in Boston. This is the most highly studied, basically, probiotics. So the mechanism, how this works, is that if you have a cytokine here, that work on basically B cell lymphoma 2, basically transcription factor. And that basically destabilizes your tight junction and you get leaky gut, okay? So if you take this probiotics, basically I work through a protein called P40, because if you take the LGG and culture it, the supernatant, you run over gel, you'll see P40 protein. That's how the active component of this probiotic work. That work on epidermal growth factor receptor, basically shut down this BCL2, basically, and you get restoration of your epithelial lining. So in the end of the talk, I want to just give you some suggestions to become a healthy individual. But what I told you today is that the gastrointestinal tract is colonized by large number of commensal bacteria, 100 trillion. We need to live in harmony with this bacteria. 20% of the vitamin level in your blood is made by the bacteria. This is the reason why every baby born in the United States, they will give them vitamin K. Why? Because they don't have any bacteria yet in their gut. They bleed if they don't have vitamin K. Bacteria produces vitamin K. Colonization occurs in early life, and I'm going to emphasize to you this critical period in life and development of babies 1 to 3 years of age. The first 1,000 days of a baby's age is very important in allowing the baby to have a healthier, basically, life in the future. Composition is influenced by diet. Diet and lifestyle dominate over genetics in shaping your gut. Microbiota provide essential signals to ensure host microbial homeostasis. I didn't show you this data, but clearly there is connection between the bacteria and between basically your immune system. Actually, a diversity of microbiome will give you a higher level of Tregs. Change in microbial composition called dysbiosis. That's the hallmark of so many diseases. And advancing understanding of microbiome will lead to new therapies and prevent diseases. Okay, so this is my suggestion to you. This is how you make your gut healthy. 1, modified Mediterranean diet with extra virgin olive oil. What's modified Mediterranean diet? White meat, white chicken meat, white turkey meat, white fish. Okay, so one important point here, that if you eat farm-raised fish, they feed it corn, like omega-6, you have to convert it to omega-3. And there are a number of people who cannot convert omega-6 to omega-3. Okay, so what you need to eat is seafood from the sea. Why? Because the fish in the sea eat algae. And what's algae? Algae is omega-3. Okay, so you need to eat modified Mediterranean diet. Why extra virgin olive oil? However, the extra virgin olive oil has to be come from the country called Jordan. And some people will know why. Because you have to have a pure, basically extra virgin olive oil, not contaminated. Okay. Decrease red meat intake, because we don't want a lot of choline and carnitine, which you need, basically change it to TMA. Low emulsifier, I showed you the data. Low artificial sweetener, maltodextrin. I want you to decrease your caloric intake. Exercise. Exercise is medicine. Okay. Increase fruit and vegetables in your diet. 5-6 servings a day. Pre and probiotics. However, take probiotics only when you are out of inflammatory state. Because look, I know for sure there was a study in Boston at Boston Children's. They have taken basically 50 patients with Crohn's, 50 patients control Crohn's without, and they gave them both probiotics. No effect whatsoever. Why? I could have told them there's no effect whatsoever. Why? Because it's inflammatory state. Probiotic doesn't work. Spice your life with little curcumin. I showed you some of our data that it's anti-inflammatory. And decrease your stress level. I know it is harder time with COVID, and we have all suffered with the stress. However, think of it this way. Stress is like clouds in the sky. The clouds are going to go away. Make your vision on the sky itself. And then feel good by helping others. We are in the medical profession, right? We are scientists, we are clinicians. We feel good by helping others. And finally, we need to enjoy life. Okay. So the road to good health is paved with good intestine. Take care of your intestine. Thank you so much for listening, and I'm happy to answer any question for you.
Oh, thank you very much, Dr. Ghishan, for this insightful lecture. And we are open the floor for questions. So if you have any question, please use the Q&A, and I will read them to Dr. Ghishan, and we have already 2 for now. So the first question, Dr. Ghishan, is, if I am predisposed to a genetic condition like high cholesterol or hypertension, due to healthier food choice and exercising, I don't have them. So will my kids still predisposed to it?
Okay, so most of the diseases we know are genetically, basically predisposed and environmentally driven. Okay, so you cannot change your genes, right? However, you can change your destiny by changing the environmental factor which affect it. Your kids may inherit basically those high cholesterol, high blood pressure. However, just like what you did, if you, especially in the first 3 years of life, 3 years of life, that's the most important, basically in allowing your child to have a diverse microbiome. Okay. So at the end of the day, diet, lifestyle dominate over genetics. Okay. So the kids may inherit genetics, susceptibility for a disease. However, If you change your environment, you change your exercise, you do the diet, which I mentioned to you, definitely that will make them in the road to good health. Very good.
Do you have a recommendation for a good probiotic?
Yes. Okay. So the most studied probiotics, especially for kids, is called Cultural, which is Lactobacillus GG. Lactobacillus rhamnosus GG. Cultural, it comes in for pre and probiotics together. Okay. For adults, there is a bifidobacterium. It's called Align, commercially called Align. Okay. Align is very good probiotics as well. In one condition, those two does not work very well. That's called pouchitis. If you have someone, the colon is being removed and we create ileal pouch, the best probiotics there is VSL3. Those you will need to ask the pharmacist. They keep it in the fridge and they basically give it to you again without prescription. But that's for pouchitis. That's the best thing. Okay. So Cultural for kids, Align for adults. These are the well-studied, basically probiotics.
What dishes do you use banana skin for? You mentioned banana skin.
Oh, not the skin. No. Oh, sorry, I don't want them to eat skin. No. I want them to eat the string. You remember, you take the skin off, and then you will have strings of the banana. So people remove these strings. These strings are prebiotics. Don't remove them. They are very good for you. Yeah.
Okay.
Don't eat the skin now.
So another question, I was curious, I mean, me too. So if the gut microbiome is unique to each individual, like a fingerprint, are cases of SIBO also very unique to each individual as it is an overgrowth of the bacteria normally present? Or do you see common strain of bacteria across many SIBO cases?
Very, very good. Very good question. Okay. So why do kids or adults, why do they get SIBO? The bacteria are present in the colon, 10 to the 14 power bacteria in the colon. Small intestine, very little bacteria. I have a slide, actually, I didn't show it to you. The stomach should have 0 bacteria. Your duodenum should have less than 10 to the 3 power. Okay. Your jejunum, 10 to the 3rd or 4th power. It's very little bacteria present there. Okay. So during lifetime of a patient, they go, let's say, and they get GI bug. They go, basically, they get acquired E. coli, enteroinvasive E. coli, they get diarrhea, they get change in the gut motility. Why? Because the infection basically changed serotonin, by the way, 95% of your serotonin, which is the housekeeping, basically hormone, is present in your gut. Only 5% present in the brain, but we don't have a medicine which upregulates your serotonin. So decrease in the motility of your gut, that result in basically less house clearing, less migrating complexes, the bacteria grows in your gut. Okay, so that's individual. It had nothing to do with the diet, no. It has to do with motility. So those individuals, we do what we call breath hydrogen testing and methane testing, because some of the patients will get constipation, sometimes will get diarrhea, depending on the bacteria in their gut. Not one specific bacteria, tons of trillions of bacteria present, okay. So the treatment actually this condition is giving rifaximin and neomycin for if it is metagenome, neomycin for constipation predominant IBS. And then after that, you decrease dairy intake, you decrease sugar intake, you decrease wheat intake, and because that will allow the bacteria not to grow. And then you take probiotics to repopulate your gut with a healthy bacteria. Okay. That's the way to do it.
So do you recommend taking L-glutamine supplements?
Okay, so the small intestine uses L-glutamine for its energy. So the energy driving basically the intestinal tract is L-glutamine. But if you eat a healthy food, which I described, for example, the meats I described, they have tons of glutamine. Okay, you don't really need to have extra glutamine. However, if you have a weight loss, for example, yes, I would suggest that you take L-glutamine, for example. That would be good for you, okay? But not in a healthy state. In a healthy state, eat healthy food and that's it.
So how effective is psyllium as a source of fiber compared with flax Is there an optimal amount of these vegetable fibers?
Okay, so we believe that 20 to 25 grams per day of fiber is adequate. You can get that by eating 5 to 6 servings of fruits and vegetables. You don't need to take fiber supplement. However, if your diet, you don't take it, then you have to take fiber. How do you take fiber? You use Metamucil, for example. There are plenty of, in the market, fiber supplements you can take. But the healthiest thing to me is eating the fruits and vegetables with their skin. That's the healthiest. But you make sure you get 20 to 25 grams per day, because your colon is not going to function without short-chain fatty acids.
Are antibiotics effective in H. pylori treatment?
Yes, definitely. H. pylori, however, the H. pylori is a smart bacteria. Tell you why it's smart bacteria. Because the H. pylori lives under the mucus layer of the stomach, covers itself by urease, making ammonia, alkaline around it. So basically, so what are you going to do? Antibiotic alone is not going to work. Why? Because antibiotic is not going to penetrate down into the mucus layer. So what you do is this. You take PPI, proton pump inhibitor, 80 milligram twice daily for adults. Kids, 40 milligram twice daily. You raise the pH in the stomach. So the bacteria thinks, oh my God, why don't I still hiding? Let me go to the surface. So the bacteria goes in surface. Then you hit it with antibiotic, okay? However, make sure that you take antibiotic where the bacteria is not resistant to it, okay? So basically flagell, clarithromycin, basically 80% of the bacteria is resistant, okay? But amoxicillin in high dosages, it's highly effective, okay? So anti-TB medication sometimes will be highly effective. So consult your gastroenterologist before you, but the trick is you take PPI to raise the pH, and then you give antibiotic. That will kill the bacteria.
So how do we adjust the modified Mediterranean diet for vegetarians and vegans? Do we need to add or adjust anything?
Okay, so whoever asked the question, I have an answer for you. Okay. So there was a paper just came out in Nature, sorry. One of my colleagues did a study in Africa. Okay, all these kids, malnourished kids, you know, they have basically malnourished kids. They have bloated tummy, they have SIBO, they tried dairy product, they tried fortified milk, for example. Nothing works, nothing works. But finally, he figured out three things which work effectively in changing their gut microbiome and improving their nutritional status. And they are what? One, they use a combination of bananas, chickpeas, and peanuts. And that combination basically changed the life of these kids. It was just published recently. It's amazing. But so what I'm suggesting to you here from this experience, that eating nuts, for example, is really, really good source of protein and fat for individuals who are vegetarians. So for flaxseed oil, for example, okay. Flax seeds, for example, okay. So fruits, vegetables, as well as source of protein, mostly from vegetables, like what we mentioned.
When buying probiotics, does it matter if the probiotics are refrigerated? I have noticed in some grocery store that they have both options.
Okay, very good point. Okay, so the probiotics, the LGG, the one I told you about. Okay, so what did these scientists do in 1989 when they discovered this? They asked for a patent, and the FDA required the following. One, that the probiotics, that this LGG, the bacteria, that survive the acid in the stomach. That's important. Second, survive the bile acid in the duodenum. Third, it attached to the enterocytes. These are the criteria. The FDA approved the drug, approved it without being refrigerated. Okay, so I don't want people listening to me to go to Walgreens or CVS and pick something on the shelf or something. No, you need to have something which is well studied, approved, basically like what I'm telling you about the LGG. That's how in 1989 the patent was given to those individual scientists in in Boston.
So do chili peppers have any influence on the health of the gut, good or bad?
Okay, so if you have reflux, don't touch them, okay? Take your Nexium before you start, okay? But in general, they are healthy. Why, for example, in Indian food, there is a lot of spices and so forth. They have less incidence, for example, of Crohn's disease compared to Indians who migrate to for Wisconsin, where they eat a lot of dairy products or whatever. Okay, so it's clear that there is benefit from having a spicy food, which allow you to have a good appetite and you eat healthier food. Okay, but unless you have reflux, if you have reflux, you need to stay away from it, of course.
So how will I know if I have inflammation before I can take probiotics? Is eating Greek yogurt enough to start?
Okay, so inflammatory states are dictated by a couple of things. One, how you feel. If you feel, for example, tired, if you feel, for example, you have diarrhea, you feel, and then biochemical tests. If CRP and SID rate elevated, basically that's inflammation. But nowadays we use what we call calprotectin, non-invasive way of testing inflammation in the GI tract, calprotectin. What's calprotectin? Calprotectin is 50% of the cytoplasm is calprotectin. But it tells me that if you have inflammatory state, because neutrophils have calprotectin, they live certain days and then they die, they release their calprotectin in the stool. Less than 50 milligrams, that's healthy. People who have inflammation will be hundreds. So combination of both.
So please discuss statins for high cholesterol versus red rice yeast tablets. And the question is, the person have diverticulosis.
Okay, so diverticulosis is really a bad thing to have, right? Okay, so you need to treat the patient to allow him to have daily bowel movement. Not a constant, because why do you get diverticulosis? Because there is weakening of the muscularis layer of the colon and therefore weak pouches. If they get infected, it's a bad. So you increase your fiber in the diet. You remember, stick with the Mediterranean diet, okay? So that's the first thing to do. But if you get diverticulitis, you need antibiotics, unfortunately. Yes.
So if the microbiome is altered by artificial sweeteners, does stevia pose the same risk?
Very good question. This has not been addressed, but my gut feeling is that all artificial sweeteners will work the same. Okay.
Eating disorders are becoming seemingly more common among college students. What step would you recommend taking to heal your gut in recovering from these?
Okay, so listen, I want to assure everyone, I have a slide, but I didn't put it in. The risk of autoimmune diseases are on the rise in the entire Western world, on the rise. Crohn's disease, asthma, for example, ulcerative colitis, celiac disease, all these autoimmune diseases are on the rise. Okay. So how can we halt basically this? My gut feeling is that diet and lifestyle is very important in shaping your gut microbiome and decreasing your risk of having autoimmune disease. So the diet I described to you is the healthier diet. Look, you open basically the Google and you will see basically hundreds of diets, from ketogenic diet to the fasting diet to this diet. Mediterranean diet stood the test of time over the last 100 years. There is no argument every time. Mediterranean diet, I can assure you, there's a study came out in Gut. 5 European countries, 1 whole year, studied people over the age of 60, put them on this Mediterranean diet, okay? And then they looked at osteopenia, osteoporosis, they looked at dementia, they looked at, everything was markedly improved. So to me, the Mediterranean diet is the best diet which is tested, okay? And change your gut microbiome, make you healthier.
Microbiome research has unlocked many possibilities and foster precision and personalized medicine. How might advances in the microbiome research serve to improve population health? And is it possible to use the microbiome research to better understand where the diversity in microbiome arises among population and improve public health?
Okay, so this is very good public health question. Very good. I think Iman will likely help me in public health issues, but no. Look, we need to change basically our food industry. We have to, as a public policy, we need to emphasize that people need to eat healthier food. I know it's expensive sometimes, and nowadays it's really tough for families. However, my gut feeling is if we change, basically move from the fast food state to a healthier state as a population. Okay, so look, so why basically people in the Mediterranean area live to 190 years old, 100 years old, and they are healthy? Okay, why? Because they eat healthier food, because they don't go basically eat fast food, you know, fried food and so forth. Okay, so my gut feeling is as a population, we in this country need to start working basically on healthier population. Personalized medicine, personalized nutrition is the future, personalized nutrition. Okay, so in the end of the day, there will be a day comes in, you come to see me, let's say as your physician, and I will basically maybe give you a, something to eat, and I measure your glucose level, I measure your fatty acid level, I measure inflammatory markers, and I figure out exactly what food you need to eat. Okay, that's in the end of the day. Microbiome story is the hot topic nowadays in science. There is no way around it. It's hot topic. Understanding your whole genome, understanding your microbiome will be the future. When it comes in to become cheaper to do and feasible, that's going to be the future.
We are at time, but I will just take this shot at the last question.
Yes.
Which say, how about beans as a protein? How do you feel about them as prebiotics?
Fantastic. Very good question. 100% agree. 100%.
So perfect. So thank you, Dr. Ghishan, for a great session and great Q&A. And I would like to thank all of you for attending our 2022 James Dalen Distinguished Lecture for Health Policy. We hope you join us again next year. Have a wonderful day, and thank you again. It was a pleasure having you, Dr. Ghishan. And if we have more questions, I will send them to you.
Thank you, Iman. And I'm grateful for you, and I'm grateful for Jim Dalen and Priscilla Dalen, because without them, I will not be here in Arizona giving you this talk.
Oh, thank you very much. Thank you to all, and have a great day.
Thank you.
Thank you.
Bye-bye.